All right. So we're going to pick up where we left off last time. If you remember, we were talking about sort of coexistence and niche overlap and competitive exclusion. And that implies that we're looking at two different species at the same time. And if you remember, we had our logistical equation for a single species, right? And that's shown here. And what we can do is we can modify this equation to take into account the impact of one species on itself, and in addition, the impact of a second species on that first species as well. And then we can do the reciprocal of that to look at the impact of species one on species two. So as you might imagine, we're going to have a couple of equations for this. Okay. So this is, the differences that you see here is now we're using N1s and N2s as opposed to just N, right? The other thing we need to think about is how these are going to interact with each other, okay? So this is just the simple logistic equation, but what we want to do is make it so that these two equations can kind of talk to each other about the impacts of each species on the other. And to do that, we use something called competition coefficients. And that's shown here by alpha. So essentially what you see, like what do you notice is different in this equation compared to what we had on the previous slide? What's the big difference here? Anybody?
Now we're subtracting, I forget what n is, but we have the coefficient, the competition coefficient multiplying the n of species 2.
Right. So
I forget what
remember,
n,
this is
what the variable n is.
Yeah. So that's a good point, Victor. Let's redefine our terms from last time. What did K stand for? Who remembers K?
Carrying capacity.
Okay, and that was like sort of the cap, how high the numbers can get. And what was N?
Population size.
Right. So when we just had one population size, then N was just how close we are to K. And remember, when N reaches K, what happens to the slope? Who remembers?
Stop increases.
That's right. So the slope is flat, right? And you're basically in stationary phase from then on, okay? But that was just when a single population was growing. Now we have to account for the fact that intraspecific competition slows growth, but now we've also got interspecific competition. So we have the impacts of species one on itself as it grows and grows and grows, but also species two on species one, okay? And so the carrying capacity remains the same for that population, whether there's just species one in there or whether you mix in another species, okay? Roughly, it can be modified a little bit and we'll kind of get into that in a second, but essentially K is going to be very similar, but how fast you approach K is going to be a function not just of the number of N1s, but the N2s, okay? Now, not all species are created equal, right? And the equation, the scenario I like to throw out for this, and it'll show up in some subsequent slides too, is, different people eat different amounts of food, right? So maybe you're a fairly slight person and, you don't eat that much, but you have a roommate, some big hulking guy that eats twice as much as you. You can imagine that if you're competing for, let's say you guys share a pizza, okay? So you've got your pizza and it arrives. My guess is your roommate is going to eat more of that pizza than you will in the same amount of time, okay? if that's the case, then we need to account for the fact that there's differences between, in this case, we're talking about species, right? So species 1 has a nutrient or a carbon acquisition, a growth rate, an intrinsic rate, but species 2 is also going to have differences in how they utilize resources. And the competition coefficient is a way for us to correct for that. So if you have 10 N1s, right? Let's take a simple example. Let's set our carrying capacity at 100 individuals. And you've got, let's say, 20 N1s and 10 N2s. If you just added them together, then we would say 20 plus 10 is 30. We're about a third of the way to 100. But what if N2s eat twice as much? Then that would actually make it so that you're really closer to N equals 40 in our original sense of how N1s would behave. Does that make sense? So what we can do is multiply, and in this case, you would multiply by a number to make N2 impacts be greater. So would alpha be greater than 1 or less than 1 in that case, do you think? If N2s eat more than N1s, consuming those resources, then do you think alpha would be greater than 1 or less than 1?
Probably greater than 1.
Exactly. And so that would be the example of where, we're essentially making it kind of like your roommate, right? Who eats more than you. All right. So what that's going to mean is that if you have the same amount of N2s as N1s, that's really like having more N2s, right? They're going to use the resources, right? And so what we're essentially doing with these competition coefficients is converting N2 to N1 equivalents, right? So we said it was 20 and 10, but if they use twice as much, then it's the conversion is 20 and 20. And so we've taken the 10 and made it equivalent to 20 of N1s. Does that make sense? Are there any questions about that? Oops. So that's essentially what's shown here, right? An individual of species 2 can become equivalent to an individuals of species 1. And the reason we're doing this is to simplify our equation, right? You guys were all taught that in, middle school math. We always want to simplify, removing variables where possible so that we can solve, okay? And so that's essentially what we're going to do here is if we can convert this and combine the N1 with this alpha N2, then we can simplify our equation. All of this will be impacting the growth of species 1. But remember, we have 2 species here. So what else do we need to do?
Account for species 2.
Right, and so how should we do that? We've accounted for them in this first equation, but what else should we do? Any ideas? We have determined the impact of species 2 on species 1, but what else do we need to do? Yeah, so Jose puts in the chat that we need to look at the impacts of 1 on 2. And so that's a reciprocal thing, okay? All right, so Robin puts in the chat, why are we dividing by the carrying capacity of species 1? Okay, good question. So let's look at this. So as n's get bigger, the numerator is going to do what? Let's say this is 100, and we started with 10 and 10, but we're making that the equivalent of, I said 20 and 10, but it's really like 20 and 20. So as we get bigger and bigger and bigger, we're going to be approaching the carrying capacity. So what's happening to the numerator then? overall. As n's go up, what happens to our numerator function?
It gets smaller and smaller.
It gets smaller and smaller. So if you have a small number on top of our carrying capacity, okay, then let's say our carrying capacity was 100, right? So initially, we have 100 minus 0 over 100. So this function is 1. But then as soon as you start adding in, this gets smaller and smaller. And so what's happening is we are creating, ultimately, this is going to go to, we can't really have a 0 in the numerator for math, but let's just set it to 1, right? So 1 over 100 is going to multiply by this. And so if we've got a 1 over 100 function here, what's that going to do for our overall rate of change, right? That's what this is, dn1 over dt, change in population 1 over time. So if this becomes a smaller and smaller number, what happens to our increase, our rate of increase?
Becomes smaller as well, doesn't it?
That's right. And does that make sense given our S-curve?
Yes.
It does, right? Because remember, we start off flat, then we have a steep trajectory, and then it flattens off. So the closer you get to the flat spot, which remember last time we showed was K, right? If we scroll back, let's go back, right? This was K. The closer you get to K, the flatter this curve gets. And that's being driven by that part of the equation. Okay. All right. And so that's why mathematically we put K in the numerator, N in the denominator. The closer you get to the carrying capacity, the slower growth will be. Does that answer your question, Robin? And so what we can do now is we can think about this in terms of like these kind of pieces of the pizza, so to speak, or pieces of the pie. So this is another way of graphically showing the resource utilization patterns, right? So species 1 in this case has a high utilization rate. And what that means is that a certain amount of resources, let's call this carbon, right? This amount of carbon will support 16 members of species 1. Species 2, on the other hand, doesn't need as much carbon, doesn't need as much food. And so in that case, they eat less of the pizza with every meal. And so ultimately that means that you can support more of species 2. And so what does that do to the carrying capacity? Is the carrying capacity for species 2 higher or lower than species 1?
Species
higher.
higher.
It's higher. And does that make sense? Right? Because if they need less food, right, then more of them can coexist on the same pool of limiting resources, right? And so this is just a nice, I like this because it's a visual representation of resource space, right? But it's kind of like a pizza, right? Or a square, right? But some people cook square pizzas, right? But if you eat a smaller piece of the pie, then more people can eat off of it. And so you can see how there's a direct correlation between the utilization rate and carrying capacity, right? But the thing to also notice here is what we really want to know is not how they behave by themselves. We want to know how they interact with each other. And so what do you take from this graph where they interact together? What's kind of the thing that stands out to you when I show you this? We haven't used up all the resources yet, but as this continues to fill in, what's the pattern that you're noticing?
the size of the portion.
Okay, and what do you notice about that in terms of like the numbers and the sizes, how they interact?
For species 2, the resource utilization is low, so the boxes are small and they're taking up less space in the whole picture.
Exactly. So are we going to be able to fit more of the 2s in there?
Yes, that's correct.
And Angel, I see that you put separation. That's just sort of an artificial thing in the sense of we're not overlapping them at this point, but ultimately the whole square will fill in. And Jose, I saw that you put the max potential for each is not realized. And that's true, right? Absolutely, that's a great point. And that's an illustration of what we talked about last time, because we said competition does what to each species? Who remembers what we said?
Decrease growth rate.
It decreases growth rate. It's a lose-lose proposition. And you can see that you're not going to get to 16 ones and you're not going to get to 64 twos. Okay. Now, alpha, remember, was our way of converting. In this case, it's the opposite of the example I gave on the previous slide. In this case, species 1 is the hog and species 2 is the light eater. So if we want to convert species 2s into species 1 equivalents, is alpha going to be greater or less than 1? Remember, last time we said alpha was greater than 1. In this case, it's going to be less than. Exactly. And so in this case, 16 over 64. So what you can see is that you put the ratio of the carrying capacities. That is how we get alpha, right? And so if we convert those N2s into N1s to simplify our equation, then what we would do is multiply the number of N2s by alpha. Alpha is 1 in 4, right? Because essentially a N2 is like you could fit 4 N2s where there's an N1, okay? And so that's going to decrease the amount. And so in this case, if you had equal numbers of N1s and N2s, when you do the conversion, you're going to be multiplying by a quarter for the N2s. Okay. Oops. So if we know what the competitive coefficient is going to do, we can essentially simplify and we could get rid of the N2 here and just make it all 1s. And if we do that, it just reverts to our original logistic equation, which is pretty easy to model, right? Now, we also said we need to do a second equation, right? What's the impact of species 2, of species 1 on species 2? So species 2 has their own growth rate. So if we ignore this for a second, right, this is just the logistic equation for the S curve of species 2. They will have their own R and their own K. And then if we add N1s in, then the question becomes, is that going to be more than 1 or less than 1 equivalent? And so we use another variable, we'll call it beta, right? We had alpha for the first equation, we'll use beta for this one. But essentially it's functioning the same way as the conversion equivalency factor. Now, earlier we said that alpha for this guy was 1 over 4. So in some cases, that would be the reciprocal, so that the impact of species 1 on species 2 would be the inverse. And so if that was the case here, what would beta be?
Will it be 4?
It would be 4. So if we have a reciprocal relationship in competition coefficients, then if alpha is 1/4, beta would be 4. And what you can see is that would drive this value up, which makes sense, right? Because every one of these guys is going to use 4 times as much. Does that make sense? Now, I say this, we have to be a little bit cautious because there are some cases where it is not exactly a reciprocal interaction. But for our purposes in this class, we can pretty much go with that. Okay. And these are the so-called Lotka-Volterra models for interspecific competition. Those of you that have taken Biology 350 may have already encountered these in those classes. All right, so let's think about the outcomes of interspecific competition. Usually, if we think back to the competitive exclusion Venn diagram that I showed, usually if they have overlapping competitive resource utilization profiles, they're competing, let's say, for food, then in most cases, one species is going to drive the other to extinction. And in fact, as I said last time, we're not going to look at the models today because we don't have time, but there's scenarios where you can model this through computer simulations, and essentially 3 out of every 4 options, one drives the other to extinction. It might be species 1 driving the other one out, or species 2 driving the first one out. But in the 4th scenario, you can end up with coexistence. And this will occur only under certain circumstances. And this is where you don't have necessarily a reciprocal relationship between alpha and beta. So what we said a second ago was if alpha was 1/4, then beta, if it's the reciprocal, would be 4. And if that's the case, almost every time, species 1 is going to drive species 2 to extinction because it uses more of the resources. Okay. However, there are certain circumstances where interspecific competition is less of a factor than intraspecific competition. So if as your population grows, that has more of a hindrance on you than the other organism does, then what we can end up with is alpha and beta both being less than 1. And it's less common, but if that's the case, that's essentially what's being modeled here. Because what that's saying is this function is less than 1, this function is less than 1. And so really what takes over in terms of the growth of these is the intraspecific or within species growth. This is what drives the population size here. And similarly, this drives the population size of this species. And then in that case, you could end up with kind of an equilibrium where they can live together. Now, let's be clear though, if they're living together, they're still unhappy because they're still competing. And so it would still be lower than their individual solo carrying capacities. Okay. This is way less common than this. In general, if you have 2 competing species, one is going to kind of bully the other one out and run it off. But there are scenarios that we won't really go into the details on, but this can exist. Any questions about that? So intra versus interspecific competition, you guys need to be able to understand the difference there. All right. Okay, so let's see if we're understanding this at all. And let's have our first poll question. So what is the purpose of the alpha and the beta in the Lotka-Volterra equations? Is it to describe the intrinsic rate of increase? Does it tell us which species has the higher resource utilization rate? Does it convert the interspecific competitive forces to intraspecific competitive equivalents? Does it tell us if there's a K-selected species? Or does it E, convert energy from one species to another? Go ahead and launch the poll. Which of these is the best answer for the role that alpha and beta are playing? All right, go ahead and log your answer if you haven't already. Okay. All right, so most people put C, but there's a lot of votes for B, so let's talk this out. So why isn't A the correct answer? What was the part of the equation that was the intrinsic rate of increase? What part of the equation was that? Who remembers?
R.
Right, it was R. We try to make these things simple. Sometimes the mathematicians are helpful, right? So R is the rate of increase. So that was not alpha or beta. Let's move on. Okay, and E was just kind of a throw-in. And D was also kind of just like. So really what it came down to for most of you, and what it should have, is the difference between B and C. So let's delve into that a little bit. So B is kind of on the right track, but it's not the best answer, right? So the alpha or the beta does reflect resource utilization, okay? So, but we have to think about the fact that it reflects resource utilization between one species and another. And so the best answer here was C, because what we're doing is we are making a conversion of the N2s into N1s so that we can simplify our equation, or vice versa. So if we look back at our equation, right? What we're doing is we're getting rid of the N2 here, and essentially we can look at this as N1 minus N1s. Because once we multiply alpha by the N2, that essentially is just another version of N1. And if it was 1/4, then it'd be a smaller number than this one probably, but no problem. And similarly with beta, we do the same thing, okay? So does that make sense to everybody? It is a reflection, the size of this is a reflection of those competitive forces, but the best answer was C. Okay. So, okay, I've been talking to you about this in theory, but let's think about this in practice, right? So what I want you to do is think about some data that we have here, okay? And so I'd like us to have a breakout to discuss how we interpret these graphs. So take a look at this. I'll just give you a little bit of setup, and then I'd like you to discuss it in your breakout rooms. So what you're seeing here is two species of paramecium, okay? So paramecium aurelia and paramecium caudatum. The top graph is what happens with respect to P. aurelia. So let's think of that as like species 1. The bottom graph is what happens with caudatum. Let's think of that in species 2. So I'm going to put you guys in your breakouts, and I want you to take a look at these graphs and try to figure out what's going on here and how to interpret this. Okay? So I'm going to stop sharing. We'll create the breakout rooms. That is not what we want to do. Okay. And let's move. Okay, all right, so let's take about 5 minutes. Go ahead and accept your assignments and talk about those graphs, and then we'll come back and we'll interpret them. All right, Caden, can you see the breakout room acceptance? Welcome back.
Dr. Dillon, I have a question.
Yes.
Just to be clear, when we say intraspecific competition, we're talking about the bacteria in itself, right? Its own species. When we're
That's
looking
right.
At
So if you have 10, let's say E. coli cells of a certain strain in a flask, they're going to eat a certain amount of food. And if you have 1 million of that same species in a flask, they're going to eat a lot more food, right? And so the S curve, the intrinsic rate of increase curve, the Lotka-Volterra, will be that typical curve for a single species, okay? But then what we're doing now is we're making it more complicated by adding in a second species, okay?
Thank you.
Yep. So let's share our screen. All right, so who wants to take a stab at interpreting what's going on in the upper curves in this top part of the graph?
I was thinking that maybe the Aurelia species is K-selected.
Okay, why so?
Well, it doesn't seem to exhibit fast growth, and it looks like it's competing well in a mixed population.
Okay. So let's maybe take a step back and just interpret what does the blue curve show versus what the orange curve show? Like what can we learn from those? And it doesn't have to be Robin, it could be somebody else.
Well, on its own, the bacteria has a higher, like, the population is a lot higher on its own.
Okay, in terms of the flat part here,
Yes, as
And
far
what
as
do we-
how high the volume can go, because it goes up like to 200.
right?
And
And
then
what did we call that again, by the way?
Carrying capacity?
Exactly. Thanks, Carlos. Okay, so I agree. So by itself, we end up with more than when we mix it together. So what is this orange one showing then? Because it's a little bit confusing, I think, when you first look at this.
Is it the carrying capacity of the species within a mixed population?
In some ways you could think of it that way, yeah. But what it is this is still the number of the same species, Aurelia, right? But it can't go as high because it is being affected by having caudatum in the same flask. But this is not including caudatum at all on the curve. It's just showing the negative impacts of that, okay? So let's come back to Robin's point though. So your point was that it does quite well competitively when it's competing with another species. And so from that perspective, I think you're absolutely right that we would potentially call this a K-selected species, right? Because it does well under competitive circumstances. Certainly does better than the other species, because who wants to interpret what's going on down below? Maybe somebody who hasn't said anything. So let's start with the blue curve. How does that compare for caudatum to what we saw with Aurelia?
I would say it uses it has a lower resource utilization, considering like how well I thought that it was able to survive a lot longer compared to Aurelia. It looks like it stops like at 22, where caudatum keeps moving. So since it's using less resources, when it's in a mixed population, it's just going to get outcompeted, which is why we see that lower curve.
Okay, so let's take a step back. You said it survives till 22?
No, Aurelia does, but you see caudatum keeps surviving past 24
Oh,
days.
so let's not be too confused by that, because actually that's just where they stopped collecting the data.
Oh, okay.
So yeah, it's not like they like disappeared after
Yeah,
that.
I thought they just
So,
like died. I was like, oh.
no,
wow.
no, we don't have any evidence to support that. I think that we'll just pretend that it probably went out as far. But so getting back to your original point though, so you're thinking that caudatum. So say again what your thoughts were. Sorry, I got
So
kind of hung
all
up on that.
yeah, all that aside, since they
Yeah.
stopped collecting the data, since the mixed population seems to like the population density seems to decrease at like 24 days, there's like heavy competition, or it looks like it gets outcompeted.
Right.
Since total population, since it's a mixed population, everyone just like hits the carrying capacity and start dying, I'm assuming.
Mm-hmm. Yeah, well, it seems like it starts at 8 days, right? That's sort of the peak that you get in the mixed population one. And then after that, it doesn't do very well, like you're saying. So it's not a very good competitor. So if it's not a good competitor, then will we call it an R or a K selected species?
Okay, if I'm not mistaken. Wait, those are the
Well, we said the
No,
first
R,
one
R,
was
my
K,
bad.
so this would be
Yeah.
R. Like maybe it grows pretty rapidly on its own, which interestingly enough, these curves are pretty similar between the two on their own, but the carrying capacity is maybe slightly lower. And I think, Victor, that you had said that the resource utilization was different, but I think we may have had it backwards, because if your carrying capacity is a little bit lower, then what do you think that means in terms of your resource utilization?
Leaves more resources.
Yeah.
So if you use more resources, you'll get to your carrying capacity at a lower number. Okay. So they're pretty similar. Oops, I don't know why this does this. There we go. So yeah, so basically they're fairly similar, but if anything, I think Caudatum uses a little bit more. But interestingly enough, when you mix it together, that doesn't mean that it automatically gets more of the pie. It doesn't mean there's a reciprocal relationship of alpha and beta and they do better. And they actually died out. So for whatever reason, and we don't know all the ins and outs of it, but for whatever reason, Caudatum is a poorer competitor. Okay, when you mix it together with Aurelia. So they both do fairly similarly. They're both very happy when they're on their own, as you might expect. But you bring them together and Aurelia grows and Caudatum ultimately goes down. So what do you think would happen, if you ran this out for another 5 days? What would happen to Aurelia and what would happen to Caudatum? Anybody?
Wouldn't Caudatum just die out eventually?
Yeah. So Caudatum is essentially going to extinction. And remember, that's what we talked about in terms of competitive exclusion, right? So if there's significant niche overlap, which I think in this case we can agree these two paramecium species were overlapping quite a lot. By the way, paramecium often are going to eat like bacteria or algae or smaller cells, right? They'll phagocytize those cells. And they're going to drive the Caudatum would go to extinction and you'd be left just with Aurelia in the long run. So niche partitioning, right, is one way that species evolve over time to decrease competition. And remember this graph? So remember we talked about how initially this might be their profile, but if you bring them together, then the resource gradient profile will change. And that's because of differential survival and reproduction of the individuals at either end of those distributions. So this was resource or niche partitioning. And so over time, that makes it so that more species can coexist with each other. How does this happen? Well, there can be a number of things. One is microenvironments, right? So when we put those paramecium into the same flask, they are forced to compete with each other. And it's kind of an artificial experiment. Incidentally, those sets of experiments were run by Gauss. You may have heard of the Gaussian distribution in statistics and things like that, right? So those are classic experiments ecologically that have been use microbes for them. But they're kind of artificial experiments that he ran. Whereas in the real world, we have microenvironments which can provide refuge and variation in the environment, which means that one organism may take better advantage of a certain microenvironment than another. And Caudatum in the real world might not die out because it can live in a slightly different microenvironment from Aurelia. The other thing is in the real world, the environment is variable. It's spatially variable. It varies on a day-night cycle. right? Which is going to be particularly important for organisms involved in photosynthesis or something that really matters in terms of having days and nights. Seasons vary, right? And so these can play a very important role. And if you think about it, there are millions and billions of species on our planet, right? That would never have evolved if we all lived in flasks. And thankfully we don't. But if the world was one giant petri dish, right, you'd have very little diversity. I mean, you'd have some. But the wonderful thing about the Earth is that it creates all of this variability. And so we have all these macro and micro habitats that have formed. We have change and variability. And there's also more than one resource, right? So if you grow those paramecium on a certain food source in that flask and they run out of food, it's over. But what if they could switch to different prey types? And what if Caudatum could feed on one prey type that's different from the Aurelia? Maybe they don't compete as much. Or maybe they compete for other things too, in addition to just food, right? And then here's a real biggie. Other biological interactions can have a huge impact. So we've talked a lot about competition, but remember, predatory interactions, grazing interactions, if you reduce the population size of the competitors, right? So let's say a virus infects half of the paramecium Aurelias, right? And it doesn't kill the Caudatums, then that might level the playing field so that Caudatum can coexist, right? So there can be other things that complicate things which allow species coexistence where it wouldn't happen in an artificial flask or on a petri dish. Does that make sense? Any questions about that? So the take home from that is that in the real world, competition, it matters and it goes on, but it's complicated. Okay. Now, as communities or species live in the real world, they also change over time. And this is a concept called succession. Now, you've probably heard this term before in other contexts, maybe like with nobility or royalty. What does succession mean in terms of like the British royal family? They've been in the news a lot this week. Who knows what succession means in that context?
The next person in line once someone passes away.
Exactly. So, you know, once the Queen of England dies, then Prince Charles will finally be king at the ripe old age of whatever, right? So succession in that context is like one person replacing another. In a microbial ecological context, it's species replacing each other. So it is the directional, continuous pattern of colonization and extinction of a habitat or a ecosystem or a site by populations. So there's a species that's there at some point in time, and then over time, maybe they go away, maybe they die out, and another species replaces them, et cetera, et cetera. So because of the dynamic nature over time of resources and competition and all these things, it's very common to see these successions. And we can classify successions in a number of ways. One type of succession, which is admittedly fairly rare, are called primary successions. And that's where you have a new habitat that forms. So for example, in a newborn baby's gut, right, that baby is born with a sterile gut. It's not going to be filled with, you know, a newborn's intestines don't have lots of bacteria in them. It's only after they start to feed or after passing through the birth canal that they become colonized and start to take on, you know, a microbiome inside their gut. The way that the species interact with each other as that colonization occurs is a primary succession. So many of these would be so-called pioneer species, things that can go in to a new environment that doesn't have other organisms there. If you rely on other organisms for your survival, you're not going to be a good pioneer species. Some pioneer species would be examples of phototrophs like lichens, right? If you go into a desert and look on a rock, you'll see lichens, and they're pretty rough habitats. But these are pioneer species that can colonize the rock, and then subsequently other organisms can come live with them. We saw that with some of the microbial mat populations we talked about earlier this semester too. And that brings up the other issue in terms of how do these successions occur over time? Why is there this change? And it can be for a couple of different reasons. One is the growth of the microbes themselves can modify the habitat and make it either more or less favorable for other organisms to come in. Or the external environment can change over time, and that can drive the change or the succession. So the latter is often a result of something like seasonality, or global change is certainly having an important impact on succession. As temperatures get more extreme, as things get drier or wetter, depending on the location, as things get warmer on average, this is going to lead to changes in which populations survive and how they change over time. The autogenic is often more of an example of where microbes themselves can make things better or worse for others. And I'll show you a picture of that in a second. A secondary succession can be an existing population lives there, but there's a catastrophe that kind of resets the clock. We think of successions as being like species 1, 2, 3, 4, and so on till the end. But maybe we go back from species 5 to species 2 if there's a forest fire that sterilizes the soil, right? Now we know that forest fires take out plants and animals, but they also can impact microbes, particularly those living in the soils of the forests. And so that can be a way to reset things. Now, I say all of this, but it's hard to identify successions in many cases. And that's partly because you do have to go back time and again, because there's a lot of temporal or time-based variability. So you'd have to revisit the same location over and over and over to see these changes. And of course, spatial heterogeneity is, even one part of a forest might be different than another part of a forest. And so is that a succession or just different conditions creating that? It can be a little bit tricky. But there are some good examples. So let's look at some primary successions. And the first one I've kind of already alluded to is microbial mats. This is an autotrophic primary succession. In this case, primary production, right? That's that shorthand for primary, is greater than respiration. And so this is going to be very common with carbon and nitrogen fixing populations. And so microbial mats dominated by things like cyanobacteria will be very common. And so this is a picture of one of my former field sites. This is an intertidal biofilm, covered these black sort of almost sticky mats that cover the surface of the sand in this bay. They would be normally just be bare sand, but these primary autotrophic organisms come in and colonize this entire area. Similarly, I've already showed you pictures of hot spring outflows where autotrophs often dominate. Now, when the water first came out of this hot spring, there wasn't this green mat that was there. Instead, over time, it grew and it got thicker and it evolved and developed through these autogenic or autotrophic successions. Now, another thing that can happen is that when you have these pioneer species coming in and laying the groundwork for new colonization. Sometimes they actually are hurting themselves by making it more hospitable for other organisms to come in and replace them. And this is called facilitation. So a primary colonizer comes in and ultimately spells their own doom by making it accessible to other competitors. So an example of that is a substrate succession. So if we break down carbon, so for example, vegetation, right? We've talked about this before. Vegetation includes cellulose, which we know is basically just glucose is strung together, but it often has harder to digest parts too, like the lignins, right? And so microbes will have both, fungi and bacteria will be able to break these things down, right? And only some organisms will have the necessary enzymes to break down lignocellulose. But almost every bacterium or fungus could break down the glucose that might result from cleaving those bonds in the cellulose and liberating the glucose. So what can happen is some primary colonizers come in, they grow on a complex substrate, but as they break it down, it becomes more available for others. And those other organisms may be able to eat those simpler substrates faster than the original colonizers do. So this is usually a case where respiration is greater than primary production. So that makes it a short-lived community because ultimately what's going to happen to all of that plant material? Why is it short-lived? If we've got the plants and we've got some primary colonizers and then ultimately more and more organisms come to use the breakdown products, what's going to be the end result of that succession where you go through different species? Assuming we don't add any more plant material to the mix.
Running out of the resource, nutrients.
Yeah, and so what'll happen to those microbes eventually?
They will become dormant or die.
Exactly. Right. And so is there anybody here who's a composter? Every year I usually get maybe like one student that does that, or do any of your families or anybody you ever know have a compost heap in their backyard? So a compost heap is essentially a heterotrophic succession, right? So if you're putting your lettuce and your eggshells and all those various things that we don't, eat, that we have as like food waste, if we put it in a compost heap, ultimately what we're trying to do is break it down, right? And we use microbes that naturally occur that do this. And at the end of composting, we need it to be a usable, sterile thing. And ultimately, it is. And the reason is that it's a thermophilic heterotrophic succession. So usually you put like some leaves or straw on the bottom, and that keeps the oxygen in there. It keeps it so it doesn't get compacted and go anaerobic. They're also recalcitrant or hard to break down, so they don't disappear very quickly. And then you're typically going to need a nitrogen source, right? And so that's why adding like eggshells are usually good for that if you have a small compost heap in your backyard. Of course, if you have a large composting facility, then you might add something like manure if you're a farmer, or sewage treatment actually often will add some raw sewage into these kinds of composting reactors that can be set up. All of this would keep it fed, right? Everybody needs nitrogen for growth, and so this keeps the microbes happy. But what happens is, usually you put a compost heap either in a container or the pile itself kind of seals itself off. And if you go back and feel it over time, it gets hotter and hotter and hotter. Why do you think it would get hot inside either the compost heap or inside a bucket? What's the source of heat there? On the screen, it says microbial metabolism, but why would that generate heat? Any ideas?
Would it be fermentation?
Well, it could be fermentation, but it can also be done due to respiration, right? And so we said we want to keep oxygen. And the reason you want oxygen there is that respiration is much more efficient at breaking down, right? Breaking down carbon like glucose, right? But why would there be heat? And let me frame it a different way. If any of you guys, it doesn't sound like you guys are composters, but if any of you are car guys or gals, why is it hot on the hood after you drive your car for 100 miles? Anybody know? Like if you put your hand on your hood right after you've driven a long distance, it's going to be hot. Why is that?
Combustion.
Right, and really incomplete combustion, right? So
Maybe because of the respiration that's going on between the like some of the microbes that might be living, they will give off energy in the form of heat.
exactly. And that's what Ashlyn, you put in the chat as well. Exactly right. Energy release. So the thermal energy is waste, right? So your car combustion is not 100%. And actually the heat that you have in your engine is bad. It's bad for the engine, but it's also bad because it's a sign that you are not using maximum efficiency in breaking down the gasoline in your gas tank. Similarly, in a compost heap, heat is released as a waste product. As they're metabolizing all of this, yes, some of that glucose is broken down to CO2, they get ATP from it, all that good stuff you guys learn in biochemistry, right? But the bottom line is they also lose some energy to heat. And if you trap that heat under a thermal blanket, which is essentially what a compost pile is, it gets hotter and hotter and hotter over time. And so this is a good thing for composting because as the temperature increases, it sterilizes the pile and eventually only truly thermophilic organisms can live there. Okay. And then even those, I mean, you don't have a lot of hyperthermophiles hanging out in compost heaps because they would have to be in your original inoculum. But once you get to a certain temperature, you've killed off all the pathogens, right? So you would never want to go digging around with your hands into your garbage, right? All of us have probably had to do that at some point for some horrible reason or another. You never want to do that because it's dirty and you wash your hands immediately after, right? It turns out you can grab completed compost. You can just grab it in your hand and it's sterile. It's safe. And that's because you've had this thermophilic succession that occurs where mesophiles that initially break down, they end up causing their own demise. And you have this succession of increasingly thermophilic members of the population. And then eventually even they die. Okay. Any questions about that?
That sterility, that's just like talking about vegetative cells, right? Like that wouldn't be including the elimination of spores.
That's a good point, Victor. So spores would definitely survive,
Okay.
right? And you're definitely going to have spore formers. And in fact, many actinomycetes, these are common soil microbes, they're gram-positive spore formers, right? So you end up with them surviving and doing very well. So it's not completely without any danger at all, but it's pretty good. All right. So another aspect of primary succession is the negative, right? So you can make it better for others to live, right? So the compost was favorable for thermophiles until it wasn't. But for a good long while there it was, right? But you can also be inhibitory, right? And this makes sense that this would evolve, because if you're an original colonizer, you don't want to you don't want to die, right? Now, of course, microbes don't have brains. They don't think of it that way. But microbes that evolve the ability to remain growing and dividing will be positively selected for. So if you're an early colonizer and you can stop other organisms from taking over, that's even better. This is less common than some of the ones I already said, but an example of this would be antibiotic production, right? So we, of course, discovered antibiotics in, sort of classic fashion by accident, right? So some fungus got on a plate, right? And Alexander Fleming noticed days later, hey, wait a minute, there's a halo around that fungal colony, and none of my bacteria that are supposed to be living on this plate are there. Essentially, the colonization by the fungus, they might have been overrun by all these bacterial colonies that grow, but because they produce an antibacterial compound, right, like penicillin, then they don't have to compete for the resources on this part of the plate. Okay. All right. So let's have another poll question. So you observe a change in algal populations blooming in a lake. So in late spring, we have a diatom, right? Remember those with the glass frustules? We saw pictures of those earlier this semester. Eukaryotic algae in late spring. But then by midsummer, they've been replaced by nitrogen-fixing cyanobacteria. Key to this is diatoms do not fix nitrogen, okay? So this is in part due to the consumption of nitrogen in the water. What kind of a succession would you call this? Would you call it a primary heterotrophic succession, an autogenic autotrophic succession, a facilitative autotrophic succession, or an inhibitory secondary succession? So let's get our poll. Let me see if I can find my poll thing. Where'd that go? Hang on. All right. So go ahead. Which of these do you think is the right answer? What kind of a succession is this? All right, go ahead and log your answers if you haven't already. All right. We are all over the map on this one, so clearly we need to talk some more about this. All right, so most of you put C, but there's quite a lot of votes for A and B as well. All right, so why is A not the correct answer? What word in there was a good clue as to why this wasn't the best answer?
Primary?
Heterotrophic?
Yeah, I think heterotrophic is the bigger, because are these guys heterotrophs? They're not, right? So all I've talked to you about is either diatoms or cyanobacteria, and those are what kind of organisms?
Autotrophs.
Exactly. Okay. So in fact, we can probably narrow that down then to B and C if we just focus on that word alone. Okay. So then the question is this autogenic or is it facilitative? And I intentionally put these here, and it's intentionally a little bit vague and confusing because I want us to really think about this. What did we say autogenic meant? What is an autogenic succession? Maybe look back in your PowerPoint. What did we say? What did you put in your notes for autogenic?
Does it talk about primary producers being the dominant? And there's more.
So autogenic doesn't have to be primary producers. So don't be confused by the auto, right? That's just self, right? And genic is, self-generating or self-producing. So what did we say so Alessandra put alter the habitat. So an autogenic succession means they've altered the habitat, okay? But it's also as a result of what? How are they altering the habitat? Are they releasing antibiotics? What are they doing to alter the habitat? Any thoughts? So in this case, what's happening is they are using nitrogen, right? So the diatoms use the nitrogen. But over time, what's going to happen to that nitrogen? What do you think? Think back to our lake. What happens in a lake over the course of the summer? If it's a stratified lake, what's going to happen to the nitrogen in the surface? Where the photosynthesis is going on. It's going to go down, right? And that is going to make it less competitive to be a diatom and more competitive to be a nitrogen fixer, like a cyanobacterium. So in this case, B was actually the better answer. So it's an autogenic succession where the activities of one organism doomed itself. They made it better for another organism to succeed them or take over time. Okay. So a facilitative example would have been something where like maybe they were releasing some sort of compound that made the cyanobacteria do better. But in this case, I would say the best answer is B. Okay. All right, well, we are over time. So I will stop there and we will finish this next time.